All right, I understand we have an hour, and I was going to try and make this slightly interactive. So I was going to take the first 30 minutes to talk, and then hopefully, I think there are what, 5 people on the call that are in the process of writing grants. I'd give people a chance to share their ideas real quick at the end for the last half hour. And you can stop me if you have questions, okay? I have no conflicts of interest. Today's objectives are to understand components of the research plan for obtaining successful grant funding, strategies for conveying your ideas, developing successful strategies for effective research approach, and maybe looking at how different plans are required depending on the different grant mechanisms. I understand you guys have had talks about specific aims in another lecture, so I will not cover that a whole lot, but it is relevant still to your research plan, so we'll talk about that briefly. So for example, if you're writing a K, then your aims and your research strategy, that together would be constitute the research plan, but technically we're going to work on that strategy, that 12-page portion today. For your R series awards, once again, your specific aims is its own page, but your research strategy is going to be that large portion of your grant, usually 12 pages, but depending on other mechanisms such as an R03, it may be shorter. Okay, so I think you all know where to go to look for these different formatting issues. So let's start with the basics. What goes into a research plan in terms of content? So I think of an hourglass model when I'm talking about giving a talk, writing a grant. Start out broad in terms of the impact and significance. Go narrow when you start talking about the details of what your plan are going to be, the meat and potatoes, if you will. Maybe it should be an inverse hourglass because it's more content. But then you go broad again at the end because you're talking about the impact that your work will have on the field. So kind of broad, narrow, broad. So first off, we're going to talk about what's the problem, right? You need to communicate why is it important, why is it interesting, what's already known and what isn't. Obviously, we all come to this process with bias. We think our problem is the most important, interesting thing in the world, but unless your audience thinks so, you're not going to get very far on a grant review. So you really need to communicate succinctly, clearly, the importance of the problem and why it's interesting. Then you get to the middle part. What are you going to do? What's your hypothesis? Why is your work innovative? How will it be accomplished? Importantly, why should you be the one to do it? And who else is part of your team and will be working with you to do this work? Finally, going back to the broad part where we're closing out, you want to say, what are the implications of your work? How will it add to the field? If you're wildly successful in this grant, what is it going to do to transform either clinical care or maybe science? Or if this is the K award, how is it going to position you to go to your next stage in career? How is it going to position you for independence? Now, within each of these boxes, the NIH also has specific criteria that they're looking for. So when you talk about what's already known and what isn't, that is your opportunity to demonstrate that you are rigorous in your view of prior research. Who's already published in this area? What work has already been done? What's good, what's bad, what's lacking. So that's where rigor of prior research comes in. It used to be go by different names, but since 2016, they call it rigor of prior research. In the green box here, when you start talking about your actual action items, this is where you want to exhibit rigorous experimental design, rigorous attention to detail, consideration of all the relevant biological variables in the study as well, whether that's a basic science project or a clinical study. So keep in mind those green boxes because they fit into the contents of the plan in terms of the qualities that you're trying to exhibit as you talk about the plan. All right. So going back to aims, I know this isn't the topic today, but aims are, I look at the research plan as essentially a blown up version of your aims. It's going to rehash what you've hopefully already articulated in your aims. So what's the problem? Why is it important? Why is it significant? What is unknown or what needs to change? What is your hypothesis? I will emphasize it should be very evident from the writing what your hypothesis is, but it's always good to just spell it right out, put it in bold. My hypothesis is. How will you test the hypothesis? And you put forth your aims. And finally, as we talked about before, you go broad at the end and talk about how will your results, how will the successful completion of this project impact the field? A couple points about aims. I tend to favor using verbs. So what are you going to do in the aims? They are action items. I'm going to examine, I'm going to perform, we are going to conduct, we are going to validate, as opposed to a passive structure. Also parallel structure. So not all grants are going to look like this, but hopefully your scientific question can be addressed from a couple different approaches, one of which might be observational, one of which might be interventional, one of which might be descriptive or qualitative. But do attempt to make your aims fit into a parallel structure. All right. You're not intended to read this. I'm showing you my specific aims page on a grant that I'm funded on right now. This is an R. First paragraph, I'm going to hit on those same topics. Why is this significant? This first paragraph just says sleep apnea is bad for metabolism. We don't know why. That's the gap. Why is that a problem? The knowledge gap prevents risk stratification in targeted therapies, and the gap and current studies are not measuring this, okay? So when you guys come to your projects later, I'm hoping that you guys can articulate literally in one sentence, what's the significance, what's the gap, okay? The next section is, what are we doing about it? What are we proposing to do about it? I wrote that our lab measures metabolism during sleep, and we found a certain phenomenon occurring during sleep. Then hypothesis, I was very explicit about that. We hypothesize that sympathetic activation stimulates fatty acid release during sleep. It can be prevented with beta blockade, all right? So that's my hypothesis. Then the aims, okay? Action words, identify, examine, right? So I'm identifying predictors, I'm examining mechanisms, and I'm examining consequences. And it's kind of repetitive, but that's the point. I want it to be parallel and simply understood from that standpoint, right? Finally, at the end is a paragraph that talks about, the implications. We may identify patients at risk and novel therapies to protect against metabolic consequences. I generally favor putting a figure of some kind under your aims page, a small concept diagram, so it's easy for the reader to follow. There has been some confusion about this on the NIH website about saying figures are not allowed in the aims. And I've talked to multiple program officers about this, and that's actually not true. There is a figure allowed on the aims page. They don't want a full, Western blot on there, but they do want, they are fine with having figures that are conceptual. Okay. So fine to put a simple concept diagram up there. All right, going back to research strategy. So here are your main criteria that the reviewers are going to care about when they see your proposal. What's the significance? What's innovative, and how are you going to approach it, right? There are two other points. If this is an R, then you're going to also be graded on the environment and on the investigator. Hopefully you're just going to, you're doing a good job of communicating who you are and where you are. And in my opinion, those should be freebies. You should be getting good marks on that. If you don't, violate any major rules and just talk about yourself honestly, you hopefully get good marks on person and environment. Hopefully, if you're writing a K or a training grant, then you have a very strong letter from someone that's going to talk about, vouch for their support of you in the division or department. Okay, but going back to the topic, significance, we hashed on this already several times, innovation and approach, and I'll go through that now. Okay, so really this is your who, what, where, when, why type of framework for proposing a grant. The why is the very first thing you're talking about. Why are you doing this? And then later when you get to approach, it's how are you doing it? If you don't do a good job convincing someone of the why, they're not going to bother caring about the how. Okay, so significance is the most important thing to talk about when you present this grant. Now, what about the when? It kind of, a lot of this is implicit. You don't have to say we're doing this now, but I think you do want to communicate that the time to do your proposal is now, because this is a pressing problem. Patients are dying from this disease, or X number of dollars are being wasted on this resource. So you want to communicate some urgency about why this should be done now. Obviously, you're the one writing the grant, but it should be communicated in some way why you are the one to do it. And hopefully that is mostly based on your credentials, but perhaps what your lab or your group has been doing to date, right? That kind of qualifies you to do the work, because you have already found that this, therefore we are going to do that next, okay? And then where, obviously, again, you're the one writing the grant, but convince people that your institution has experience and the qualifications to support for you to do this work, okay? So keep in mind all the W's, who, what, where, when, why, and then the how is the nitty gritty of what you're going to do later. Okay, a few more details about the strategy. When you get to your research strategy, just a few points. Really focus on how you're going to improve the current status quo, whether that's technical knowledge, capability, clinical practice, and be relevant to the topic area. So I've read several grants where it's thorough, but a lot of what's being discussed is not even relevant to the grant. I mean, it's not your chance to show off how much you know about this topic at large. It's your chance to demonstrate your knowledge of the specific area that you are looking at. So please be relevant to what you are talking about in the grant. Innovation. I think this is self-explanatory. How are you being innovative? Now, your project could be innovative solely on the basis that the concept is new. No one's thought of this before, right? It could be innovative on the basis that you are going to use this shiny new technique or device or drug, right? That's innovative as well. Maybe it's innovative because everyone else in the world is using this metric to measure something, and you're going to convince people why they should be using a different metric. So those are potential innovations. This comes down to style. I actually think it's worth spoon feeding people the innovation in almost a bulleted list. Like, these are the innovative parts of my proposal. And you could actually put a bulleted list. These are the innovations in this study. Feed it right to them, right? Approach. Okay, this is self-explanatory. This is what you're going to do. How are you going to do it? Why, are these methods important to do? How's the data going to be analyzed and interpreted? Statistics are important as well. I know that the audience is very highly varied. If you're proposing a basic science study, they may not really care as much about the statistics, for example, but do not make the mistake of saying, well, this is a pilot study, so we're just gonna, and you can tell that someone has spent 2 sentences writing a sample size. That really needs to be carefully discussed with a statistician and worked out for your study. All right, so this is a bird's eye view of my proposal. You're not expected to read it. I'm going to just show you the sections that I put in. So innovation, remember that in the first opening salvo of your project, talk about the rigor of the prior research. So what has been done in the study, what has been done, what needs to be done still? What are the weaknesses and gaps? Innovation. So notice here I put number 1, we challenge the basis for this. Number 2, we pioneer a precision medicine approach. Number 3, we apply innovative stable isotope techniques. So I'm telling them what's innovative. And guess what? When someone is reviewing this and you're the reviewer and you've read like 10 grants, you basically, and you have to actually put your assessment of the person's ideas in writing to the study section. I've seen people just copy paste my innovation and put it in there because you've handed it to them, right? So I really think that there's some places where you just show them why you're being innovative. For another area that we'll get to later when we talk about consideration of relevant biological variables. Rather than make them hunt around for that, you have a sentence that says in bold, consideration of relevant biological variables. We are going to look at gender or sex as a variable in this study because you're going to just hand that to them, all right? Don't make them hunt for it. Okay, let's get to the approach. This is where you're going to tell them what you're doing. And remember, It's all about first setting them up for caring about why. Now, I'm going to be honest with you and vulnerable, even though I was selected to give this talk, because I've had my share of successes in grants. I've had many failures. The last 4 R01s I've submitted have not been funded, okay? And some of them are because it fell down on approach. They said, okay, I got 1s and 2s on significance. I got 1s and 2s on everything, but then approach was a 5 or a 6. So it does matter, but you do have to win the battle first about saying why the work is important before you get into the approach. All right, so in writing your approach, you do want to structure it. It's kind of like a repetition of the aims again. There are different styles of doing this. Some people kind of lump everything together. I think the best way is to say, aim 1, here's my approach. Aim 2, here's my approach. Aim 3, here's my approach. And every section is the same template, okay? You repeat your rationale. Why are we doing the same? You go through what you're doing in the experiments. You emphasize rigor in what you're doing. And once again, I think it's important to, I don't think it's being condescending to say, this study is rigorous because we are doing a blank. Double-blind, placebo-controlled, randomized study, crossover design. We are doing, 22 replicates of this, or, just tell them, why is it rigorous? But don't make them hunt for that, okay? Pitfalls are important. Pitfalls and alternative strategies. This is an opportunity to exhibit knowledge and forethought about what could go wrong, okay? Especially in a K where your training is really the paramount issue, right? Your hypothesis could be dead wrong, but will all the work be for naught when you're finished? Hopefully there's something that could be still gleaned from what you've done, something that you've learned despite what happened. And so I think it's important to exhibit just a few things that the top things that you think are like someone reading your grant would say, have they thought about this? And keep it to, obviously that list could be infinite, but keep it to the most likely issues. I had a study, for example, where it was a randomized crossover trial that I was proposing. And frankly, I wasn't sure if there should be a washout period between the 2 arms. All right, I went back and forth about that a long time. And I could see why I might get dinged by saying there's going to be a washout period. I could see how I could dinged if I didn't put it in. So I put it as a pitfall. I said, we understand that a washout period might be helpful, but this is why we don't decide to do it this way. And if we find a carryover effect, they will implement a crossover, will implement a washout phase. All right, so show them that you have thought about the issue. It almost gives you, it's not going to give you a pass on a glaring problem, but at least they'll kind of sit back and say, okay, this investigator has thought about that problem, right? And then again, analysis of the data and justify the sample size. Why are you picking the number that you did? It doesn't have to be powered. This getting into a detail here. A study doesn't have to always be powered to prove your hypothesis, right? You want it to have powered a sample size to get to your goal. Now, if your goal is to do a pilot study that will one day address your hypothesis, then you should cater your sample size to that goal, not to the grand R01 that has to be done 5 years later, which is going to need 10 times the sample size. Okay, so just be realistic about your sample size goals and where you are in the process of proving your hypothesis. Okay. All right, so this is very small, but I'm showing you just to give you a bird's eye view. So here's my approach. I'm organizing bullet points, or I'm organizing sections by specific aims. And remember, this is your opportunity to talk about the rigor of your experiments, okay? So preliminary data could also be put into this first section of the approach, where you talk about what was done relevant to this aim. I've seen different methods of doing this. Some people put a whole section of preliminary data before they get to approach. Some people put all the aims and lump them together and say, here's the preliminary data for this whole project, and then you start talking about each aim. Another method, which is what I did here, was you could do aim 1, preliminary data or preliminary studies for aim 1, and then you go through that, and then you go through aim 2 and preliminary studies for aim 2. Okay, I don't think it's a right or wrong approach. It probably depends a little bit on the concept that your grant is about, whether that's warranted. Okay, certain studies, as you probably know, certain grants do not require preliminary data. R21s, R03s, as an example, they're acknowledged to be high risk, high reward type of designs where you don't have data to display. Okay. Okay. Yes. A word about study team. So there are a couple places in your grant, such as the bio sketches, such as the letters of support, where you will be spelling out who's doing what. If you have the space for it, I think it's helpful to also put a blurb about who's doing what in your approach as well, because it will re-emphasize why you have the team that you've assembled and what they're all going to do. Okay. It is a lot of work to read through these grants. So sometimes going through 4 bio sketches, you can read all of them and say, wow, these are great people, but you still don't really know what they're doing. So I do think it's helpful to reiterate that potentially somewhere in your approach. All right, so here, this is a couple pages, which is aim 1. I went into the intro and rationale for aim 1. I talk about the design, the procedures, the analysis, the stats, and then the pitfalls and approaches. All of that pertains to just aim 1. Okay, then I came to aim 2, and I did that same process. All right, so this is one method to approach it. Just divvy up your document into aims. Let me pause. Any questions on that so far? Okay. All right, rigor and transparency are sometimes a little confusing. This used to be called scientific premise in 2019, and then NIH decided that was a little confusing what is meant by premise. So now they call it rigor and there's 4 criteria where they want to show that your grant is rigorous and is transparent. So number 1, rigor of prior research. We discussed that earlier, going through the prior literature to talk about what is known and what's not known, and what are the gaps. Okay. Then there's the rigor part of the experimental design. Third is the consideration of relevant biological variables. And 4th is authentication of key biological or chemical resources. We'll talk about each of these briefly. Okay. So number 1, rigor of prior research. Describe the scientific premise. That's that word again that they used to use for the proposed project, including consideration of strengths and weaknesses of published research or preliminary data crucial to support your application. Okay, and then talk about the gaps and weaknesses in the existing literature. What's the problem? Why does your work need to be done? An interesting thing that was actually put on the NIH website is, as an example of this was a paper that said in RCTs that were published, many of these trials cited very few studies in their discussion of the results. So that's a lesson of what not to do, right? You want to really be thorough in talking about what has been done in this space before. Likely someone on the study section knows as much or maybe more than you do about your area. And you don't want to come off as sounding ignorant about, oh, someone already did this study 20 years ago, or this hypothesis has already been proven or disproven. So make sure you go through that, do your due diligence in the rigor of the prior research. Number 2, rigorous experimental design. So could someone read through your project and say it's rigorous? That's somewhat subjective, but I think we all know there are key elements of a design of a study that make it more rigorous in terms of the design, the number crunching of stats, and the way that the trial, if this is a clinical study being designed, it could be using X number of replicates or standards in the analysis. Is your study going to be blinded? Even if this is a basic science study and you're doing like mouse studies, who's going to be reading the data? Or is that person who's going to be interpreting the results? Is that person blinded to the arm that you're in, and do you have very clearly defined inclusion and exclusion criteria? Is there an easy way to say, here's the pipeline going from raw data to the finished product? All right, so remember, what are our synonyms for rigorous, right? Meticulous, exact, stringent, severe, strict. So could someone look at your proposal and say, this is a rigorous study? All right, consideration of relevant biological variables. This is basically the NIH trying to make sure that your study does address important things such as sex in the design and analysis, right? And there should be a strong justification if you're not going to be doing that. Obviously there's some conditions that only affect men or women, and that it should be self-evident why you're only choosing to study one sex or one race or one experimental condition, but otherwise it is expected that you will look into those other variables. In the sleep world, we're also going to be dealing with other variables like circadian rhythms, light dark cycles, BMI, seasonality, right? Make sure that you are showing consideration of these biological variables, right? If you're working with cell lines, you're working with animals, who are your vendors? What are your strains of mice? What are your reagents coming from it? Understand that those could also present some variability. Authentication of key biological and chemical resources. So that's basically what are the raw ingredients that you might be doing. This applies a little bit more to bench work, like if you're going to be studying a certain gene, or you're using antibodies in your work, where are you going to be getting your reagents? How do you know that they're stable? How do you know that they're reliable, right? So just make sure that you lay that out as well in your approach. Okay, so this is just to kind of rehash. Where in the grant application should applicants address the 4 focus areas? So rigor of prior research goes into your significance section, right? That's the opening part. Talk about prior research. That's the rigor of prior research. When it comes to scientific rigor and biological variables, that's in the approach section. That's where those elements should be evident. The authentication part is actually not a scored item. It does not contribute to your score, but it is still a required element in the grant application. All right, what's the difference between rigor of prior research and significance? So rigor of prior research is a retrospective consideration of the foundation for the application, and it is part of the significance. Okay, so remember, your significance is the broader discussion of the importance of the work. Rigor of prior research is just one element of significance where you get into foundational knowledge leading to this project. All right, a quick thing about formatting. Make sure the fonts are the right size. Make sure your fonts, particularly in figures, are not too small. Now, the NIH does say as long as it's legible, it doesn't have to be 11 point. But it is highly irritating to a reviewer to not be able to read your y-axis on a figure. And that shouldn't be the reason that you get dinged, right? So just make sure your fonts are of adequate size. Use sans serif. So as an example, that would be like Arial. No little symbol, no little lines at the end of the fit characters. That's a serif font. So you want sans serif. And then just look very carefully at the requirements of your grant on spacing and so forth. It is not good to have a grant that is just jam-packed with line after line of text. There should be an ample amount of white space. There should be figures whenever possible. Put a figure in instead of a table. It communicates a lot more information, and it's much more pleasing to the eye to have figures on your grant. Okay. Also, avoid too much jargon or abbreviations. It's very, again, annoying to be 10 pages into your research plan, and then I have to go back and flip 4 pages back to find where you define what ARTXW meant. Like, it should, if it's only going to be used 3 times in the application, don't abbreviate it at all. Otherwise, just make sure you are very clear on when you define all your acronyms. Okay. A word about pitfalls and alternative approaches. Justify what you did. Okay. So you have to thread this careful needle. You have to talk about why you're taking the approach you did, but then you have to be humble enough to admit that what you picked could be wrong or might lead to problems. So by all means, justify this is why we're looking at it this way. But okay, the null hypothesis could be the outcome of this grant. We all need to be well aware that that is a likelihood that things will not work out as you hope they would. But this is also your opportunity to talk about, regardless of the outcome, this project is critical because, okay, and it could be because it fulfills certain training goals. It could be because it will still move the field forward. A negative study that is well done still needs to be done. We don't need, that's the problem with journals nowadays. They're looking for highly significant findings of a p-value rather than a well-done study that may still be negative, right? Anticipate what viewers may ask, and in terms of the pitfalls, as we discussed before, that could be at the end of your application. It could be pitfalls about the whole thing, or it could be pitfalls for aim 1, pitfalls for aim 2. I don't think it matters. I think it's dependent on the character of your grant. Timeline, that's usually like the last thing I have in a grant. It's definitely needed for a clinical trial. It's less important for other work, whether it's observational or bench work or epi, but it's still good to put it in. So because it shows that you've thought about how your 3 years or your 5 years or your K99 and your R00 phase, or how you're going to be dividing up that work over time. Okay. Know your audience. I think this is self-explanatory. I'm going to skip through that. Let me stop there because I think that's all I had to say in terms of, quote, didactics.